Hello, I'm Cole Younger, and this summer I worked in the Largent Milnes lab, which studies the physiology behind headache and migraine. For context, headache and migraine are common debilitating conditions that stem from a dysregulation of the endocannabinoid system, the brain's pain modulation pathway. The periaqueductal gray, or PAG, is a vital component of this pathway that influences headache initiation and persistence. Furthermore, astrocytes, regulatory cells within the brain, are found to play an active role in sustained neuroinflammation associated with headache. Previously, my lab developed a protocol to isolate and culture astrocytes from mice spinal cords. This summer, I adapted this protocol in order to isolate and culture astrocytes from the PAG of mice, which will allow us to study these astrocytes' behavior and health in vitro or within a controlled environment. Additionally, we investigated CB2, a vital endocannabinoid receptor, through both behavioral testing and astrocyte cultures of CB2 lacking and typical mice. Through our first isolation, we found that 1 PAG is sufficient for cell cultures. Then, through behavioral testing, we discovered that CB2 is vital for pain management in female mice. Finally, by applying our protocol, we compared typical and CB2 lacking astrocytes through both cell quantification and imaging. Through imaging, we found that the typical astrocytes were more developed. However, the adherence of microbeads to cell surfaces hindered exact quantification and imaging. Ultimately, our experiments illustrate that our cultures can be used to compare astrocytes across genotypes, and in the context of migraine and headache research, our cultures can be used to investigate the effects of medication overuse headache-inducing drugs such as opioids and triptans and how they affect astrocyte health and behavior. Thank you.